Hi, my name is Andrew Phillips. I'm head of the Biological Computation Group at Microsoft Research in Cambridge in the UK. And today I'm going to talk about some of the work we've been doing in computer software for programming life. So we can think of living cells as highly sophisticated computational machines, constantly processing information from the environment to survive, grow, and reproduce. And we can program cells by inserting new DNA that can code for new behavior. And the ability to program cells could essentially transform our ability to address some of the many challenges we face in society today. And remarkably, we can think of cells as essentially the highly efficient computational devices. So the DNA inside the cell can essentially code for a gigabyte of information in less than a millionth of a cubic millimeter. And the cells sort of have their own power supply, they're massively parallel, they repair themselves, and they essentially completely reproduce themselves. So if we could program cells, we could address problems in health, food, and energy. For example, cells could be programmed to control the spread of viruses and cancers, to feed the planet in more sustainable ways, and to essentially produce renewable energy, such as by converting sunlight and carbon dioxide into biofuels. And in developing these technologies, we could gain a deeper understanding into how life itself works. In spite of this promise, there are still many challenges to overcome, including how to compile a high-level cell programming to low-level DNA machine code that functions as expected inside the cell. And as cell programs become more complex, software for programming cells will become essential to addressing these challenges. So in my group, we've developed a programming language for programming cells. Essentially, the programmer writes a high-level description of the desired behavior of the cell, and the software is used to analyze its behavior, simulate the behavior, and generate the corresponding DNA code that's needed to be put inside the cells to achieve this behavior. As a simple example, we can program a receiver device that produces a receiver protein that binds to a signaling molecule after it crosses the cell wall, and then activates the production of GFP. This is just an example. And so we can simulate the behavior of this device in a colony of cells in a petri dish. As the signal diffuses from the center of the dish, the surrounding cells take up the signal and then fluoresce in response. And we can calibrate this simulation results with experimental data from the wet lab. And using these basic techniques, we can then eventually program more complex functions inside cells, such as cells that communicate with each other to form complex patterns of gene expression that are robust to perturbations. Using similar techniques, we've also developed computer software for programming DNA circuits. So the idea here is that a programmer writes a high-level description of the logical structure of the circuit, and then we can compile and simulate this behavior on a computer before outputting the corresponding DNA sequences. So as a very simple example, we can program a logic gate, which takes two DNA strands as input and produces an output only if both strands are present. And these types of circuits could be inserted into cells to, for example, detect the presence of cancers and in the human body. And if, for example, both cancer markers are present, the cell death is triggered. This software has so far been used to stimulate and analyze some of the most complex DNA circuits implemented to date, including a circuit for implementing the square root of a 4-bit number in DNA, and also an associative memory in DNA, and some of the references are below. So just as high-level programming languages revolutionized the computer industry, we anticipate that high-level languages for programming cells could pave the way for a new wave of biotechnological innovation. Thank you very much.